And if you guys have any questions, just place it in the chat. But we're going to start by talking about Module 2 and why you don't see many chapters and their PowerPoints there. That is because I'm all out of space in Canvas. This hasn't happened at other schools, but for Long Beach, I've run out of space. So what I'll have to do is take out the PowerPoints from a previous module so I can get space in Canvas to upload this module's PowerPoints. Tonight, we'll be going over Chapter 9, which is about skeletal muscle contraction. And we're going to be going over PowerPoint A, which I have not uploaded yet. And PowerPoints B and C will be very similar to what you cover in lab. So kind of tonight, we'll try to get through Chapter 9. If we need to, we'll use Thursday to finish Chapter 9. But that kind of takes us through. So here's your second case study, the discussion post. There will be a quiz probably in a week or two that I'll explain, make an announcement about that due date. You can see where the exam will be. And in this module, remember there will be 2 lab exams throughout the semester, and there will be one at the end of this module, and it will include information from labs 1 to 5. And then there's just 2 labs to get through this module because we'll have an exam at the end. The Lab 4 will be working with Physio X, which uses the MyLab and Mastering, and I'm going to show you how to get into that. Lab 5 will be working with Labster again, but Lab 4 is on skeletal muscle physiology, and it's exercise 2 in Physio X, and you'll have to complete all of the activities for that. I think there's 7 different activities to go through, but I give you 2 weeks to do this lab. I think that's in 2 weeks, right? October 11th, a little less than 2 weeks away. So to submit it, you'll need to submit all the PDFs of each lab activity report. And I'll show you right now how to get there. So again, this is the most recent lab. You'll go into MyLab and Mastering. Oops, not grades. Sorry, guys. I don't want you guys to see your grades. There we go. Computer's being a little slow. So we'll go into the MyLab and Mastering. You'll open MyLab and Mastering. Hopefully you guys are familiar with how to get here. And then you'll go to the study area. Can everyone see so far? You'll launch the study area. And then you'll go into Physio X. And you'll go into the exercises. And the exercise that we'll be focusing on for the next 2 weeks is exercise 2, the skeletal muscle physiology. And I'm going to ask you guys to do all 7 activities. And I'll take you through kind of what a typical activity looks like. So once you do, and each activity doesn't take that long, but I give you 2 weeks to do this. Each activity takes you through a series of questions. You have to read through them, kind of a pre-introduction, then there's a pre-lab quiz that you go through, and then eventually you get to the experiment itself. And the experiment for this one involves stimulating a muscle and seeing the force of that muscle from a stimulus. So it's actually a great lab simulation to describe all about skeletal muscle physiology. So you work through, and then at the end of the lab experiment, and I don't know if it'll let me get through all of it. I'm just submitting, and you'll go through and do a post-lab quiz, and then you'll answer these questions. And you will want to type in an answer for this, because if you get the quiz questions wrong, I'm not too worried about that. But here I will be checking your answers in the review area. And then eventually at the end, you can submit and you get this whole lab report that you're able to save as a PDF. So this is the printable savable version. So you just click that, you can save it as a PDF, and then you can upload that PDF to the submission area in the module for this lab. And you'll have to do that 7 times because there are 7 activities associated with this exercise. Does that make sense? Questions on that? So again, the next lab we're doing is in Physio X. To get to Physio X, you have to open up My Lab and Mastering. And then once that's open, you click on Study Area, you click Launch the Study Area, and then you click on Exercises, and that'll open up all the exercises in PhysioX. We'll be doing several of them throughout the semester, but this one we'll be doing is this exercise 2, skeletal muscle physiology. You'll work through all 7 activities. You'll save each as a PDF, and then I'll show you where to submit those again. So we go back to the module. I have the submission option right in the module. So right in module 2, here is the Lab 4 PhysioX submission option. So you'll just click on that and you'll upload the file, your 7 PDF files to submit them. If you know of a way how to convert all the PDFs into one file, you're free to do that. But otherwise, I'll just look at all 7. If you know how to combine all 7 into one PDF, that would be great. But if you don't know how to do that, that's okay. And I'm sorry I don't have instructions for you how to do that right now. If anyone knows how to do that and wants to put it into chat, or I'll start a discussion for this lab, then you can feel free to share how, because I think there is a free way to convert all 7 PDFs into one. And if anyone wants to volunteer to explain that to the class, that would be a huge help. But otherwise, just you can submit all 7 separately. Yes, it'll run a lot better than Lapster, Aaron. It won't use as much power. It's probably the way we're going to be doing a lot of the labs for the rest of the semester, because you won't really have any issues with, like you did with Lapster. It'll run a lot more smoothly because it just doesn't require as much power. Okay. Questions on the lab. You have 2 weeks to do it. I'm giving you ample time. Start it now. It's all on skeletal muscle physiology, but it will take you some time to complete that one. All right. So with that, if you're just joining us, we are in part A of chapter 9. Parts B and C will be kind of what you cover in your lab, but part A will be on the sliding filament model and skeletal muscle contraction. The first, and I will upload this PowerPoint once I delete some old PowerPoints off module 1. I've run out of space in the LLBCC canvas. The first couple of slides take you through a review of skeletal muscle anatomy and the different types of muscle tissue and the comparisons of them. And I'm going to have you guys review that on your own, because I'm hoping you maybe don't remember it from anatomy, but it should be a review. The muscle functions, and then a little bit more about the skeletal muscle anatomy of a skeletal muscle. Where we're going to start is kind of at the microscopic level, which isn't too much further down, but we're going to look at the microanatomy. Can you guys all see my PowerPoint? 9.3. Can't see anyone. We can give a thumbs up. Okay, I'm just going to assume you can see my, thank you, Nina. Okay, I'm going to assume you can see it. Okay, so let's take a look at the microanatomy of a muscle fiber, because this will take us into the physiology of how muscles will contract. Skeletal muscle fibers are long cylindrical cells. So when we talk about a muscle fiber, that means a muscle cell. The sarcolemma is similar to the plasma membrane. So it's just the exterior border of that muscle cell. The sarcoplasm is like the muscle fiber cytoplasm, which contains many glycosomes for glycogen storage, as well as myoglobin for O2 storage, because oxygen will be necessary for muscle contraction. These are some modified organelles within a skeletal muscle cell, and we'll be going over those. Myofibrils are densely packed. They're rod-like elements. Features of a myofibril is that there are striations in them, there are sarcomeres, there are myofilaments, which are actin and myosin, maybe that rings a bell, and we'll look at some of the molecular composition of those myofilaments. So this is a look at a part of a muscle fiber showing the myofibrils. One myofibril is extended from the cut end of the fiber. So you can see this myofibril being pulled out here, and it will be made of myofilaments, and it'll be arranged in striations and also sarcomeres, which is the contractile unit. So the striations are just the stripes formed in the myofibrils, and they are formed from repeating series of dark and light bands. I will expect you guys to know the names of these different types of bands. The A band refers to dark regions. The H zone refers to the lighter region in the middle of the dark A band. The M line is the line of the protein that bisects the H zone. The I band is the lighter region, and then the Z disc or Z line is like a coin-shaped sheet of proteins on the midline of the light I band. And this is a look at the striations of skeletal muscle fiber, and you can kind of see the dark and light bands making up those different striations. In the fiber itself, the nuclei are pushed off to the exterior kind of sides of the muscle fiber. The sarcomere is the smallest contractile unit of a muscle fiber, and it will span from Z disc to Z disc, and individual sarcomeres will align end to end along the myofibril, kind of like boxcars of a train. So this is showing you the sarcomere, and it will kind of span the myofibril from Z disc to Z disc. And you can see here where these bands and zones are referring to in the myofibril. So again, a sarcomere is one contractile unit of skeletal muscle. And what happens during muscle fiber contraction is the sarcomere will shorten. And when it shortens, the muscle pulls and it contracts, and that causes movement of whatever area of the body it's in. So the lengths of the bands and zones, some of them might change. The actual myofilaments within these sarcomeres, the length of the myofilaments is not changing. The sarcomere itself is just shortening when those myofilaments slide past each other. And we'll go into a little more depth how that happens. So these myofilaments are actin and myosin. Actin are thin filaments, and they will be anchored to the Z discs. Myosin filaments are the thick filaments, and they are connected at the M line. And the sarcomere cross-section will show like a hexagonal arrangement of one thick filament surrounded by 6 thin filaments to kind of make a hexagon-like shape. So here's a look again, Z disc to Z disc, and this gives you a more detailed look at what a thick filament looks like and a thin filament looks like. The thin filaments will be attached to the Z discs, and the M line goes right down the middle of these thick myosin filaments. The thin filaments are in blue, they're thinner, they're globular proteins that are wrapped up around each other in a helical-like structure. And the thick filaments have little heads coming off of them, kind of like golf club heads. And what happens during skeletal muscle contraction is all of these like heads from the myosin will reach up and attach to the thin actin filament. And all of those myosin heads will pull the actin filament along. And as those myosin heads pull the actin filament, it causes the sarcomere to shorten, or the actin to slide past the myosin. And that is the shortening of the sarcomere that causes the contraction of the muscle. More molecular composition of the myofilaments. This describes what makes up the thick filaments in general. It'll have a tail and then a head, a globular head. And that globular head is what will link the thick and thin filaments together, forming a cross bridge. And that's just the name for the connection piece between the thick and the thin filaments. Myosins will be offset from each other. So there will be a staggered array of heads at different points along the thick filament, just to attach to different areas of the actin. The thin filaments then, actin is a polypeptide, so many peptides made of kidney-shaped G-actin subunits. They're linked together to form a long fibrous F-actin. Basically, these F-actin strands are twisted around each other to form a thin filament. We have 2 proteins that are also involved with actin, tropomyosin and troponin. And these are just regulatory proteins that are bound to actin. And I'm going to show you what those look like now. First, we'll look at thick filament. So this is the molecular structure of a thick filament. You can see here's one myosin molecule and how several myosins are put together to form this thicker, thick filament itself. You can see the different heads at staggered positions. And then the thin filament is made up of these like double wound strand of beads or globular proteins. These are the actin subunits. On those actin subunits, there's a little depression for the myosin binding site. And that little depression is where the head of the myosin will reach up and touch the actin. Now we'll talk about these two regulatory proteins, tropomyosin and troponin. And if you remember anything from tonight, try to get in your head the differences between tropomyosin and troponin. Tropomyosin is the spaghetti-like structure that covers up all of the myosin binding sites on the actin. And troponin is just kind of a singular protein that will help the tropomyosin move off of those shapes off of the binding sites when the myosin heads are ready to bind. So again, the tropomyosin are the spaghetti-like pieces. They cover the myosin binding sites. And the troponin is just a regulatory protein that will help push the tropomyosin off of those binding sites. So those are the two regulatory proteins, tropomyosin and troponin. And this is a thin filament actin. Other proteins, I'm going to kind of go over this for now. There's other proteins that help form the structure of the myofibril. I'm not going to ask you guys too many questions about this. However, dystrophin will link the thin filaments to proteins of the sarcolemma, which is like the plasma membrane. And I think we're going to talk a little bit about Duchenne muscular dystrophy here because this is a clinical kind of connection. It's always fun to relate this to real life. Abbreviated DMD, it's most common and serious form of muscular dystrophies. It basically destroys muscle. It appears during childhood. The average lifespan is maybe 20s, maybe into the 30s with supportive care, but basically it's just the muscle destroying itself and there's no cure for it. It's inherited as a sex-linked recessive disease. So that means that it's only in males. And it will appear between 2 and 7 years old if a boy becomes clumsy and falls frequently. What else? It will progress from extremities upward, finally affecting the head, the chest muscles, and cardiac muscle. So someone would just stop breathing or their heart would stop beating. It's caused by a defective gene for dystrophin. And these were one of those proteins. That's why this was one of the extra proteins I mentioned. It's a protein that links thin filaments to the extracellular matrix. The sarcolemma of these Duchenne muscular dystrophy patients will tear easily, allowing entry of excess calcium. And if we have too much calcium in your muscle fibers, that will damage the contractile fibers. So that is Duchenne muscular dystrophy. Here's a boy with Duchenne muscular dystrophy trying to work on some exercises to increase strength. Okay, so back into some of the anatomy of the muscle fiber. The sarcoplasmic reticulum is a network of smooth endoplasmic reticulum tubules that surround each myofibril. Most of them run longitudinally, and they are involved in terminal cisterns, which form a perpendicular cross channels at the AI band junction. The big thing about the sarcoplasmic reticulum that I want you to remember is that it helps to regulate intracellular calcium levels. So your sarcoplasmic reticulum stores and releases calcium, and calcium is required during muscular contraction, and calcium is always stored in the sarcoplasmic reticulum. T-tubules are a tube formed by the protrusion of the sarcolemma that travels deep into the cell interior. It increases the muscle fiber's surface area. It allows the electrical nerve transmissions to reach deep into the muscle fiber. So a T-tubule is kind of a deep pit within the muscle fiber where that electrical nerve transmission will travel into the fiber, just to allow that transmission to get into the muscle. The tubules will penetrate the cell's interior, and a triad is the area formed from the terminal cisternae of one sarcomere, a T-tubule, and a terminal cistern, or kind of outpocketing of a neighboring sarcomere. This takes you through the triad relationships. The T-tubule will contain integral membrane proteins that will protrude into the intermembrane space. The sarcoplasmic reticulum cistern membranes also have an integral membrane proteins that protrude into this intermembrane space to help control the opening of those calcium channels because the sarcoplasmic reticulum will store calcium. So simply put, when an electric impulse passes by, so when a skeletal muscle fiber is electrically stimulated, the T-tubule proteins will change shape, causing the sarcoplasmic reticulum proteins to change shape, and that will cause the release of calcium into the cytoplasm of that muscle fiber. And that's what we want. We want an electrical impulse to travel into our skeletal muscle cell in order to change this protein shape on the sarcoplasmic reticulum to get the calcium to be released, because we need calcium release in order for contraction to occur. So this kind of is a picture of what I was just describing. So here's the triad made up of the T-tubule and the terminal cisterns of the sarcoplasmic reticulum. Here are the tubules. So the sarcoplasmic reticulum is in blue and the T-tubule is in light blue. So what'll happen is that electrical impulse travels down the T-tubule into the muscle fiber itself, and then it will change proteins on the sarcoplasmic reticulum in dark blue, and that will cause the sarcoplasmic reticulum to release calcium. Contraction of skeletal muscles then is the activation of a cross bridge or a connection between your myosin head and your actin. This will generate a force when shortening occurs. Tension will be generated by the cross bridge on the thin filaments exceeding the forces opposing the shortening, and the contraction of a skeletal muscle will end when the cross bridge becomes inactive. In a slightly relaxed state, your thin and thick filaments will overlap. The sliding filament model of contraction will state that during contraction, thin filaments will slide past thick filaments, causing the actin and myosin to overlap more. It's important to note that neither the thick nor the thin filaments change their length. They only change the amount of how they overlap. So they're sliding past each other for the sarcomere to shorten. I'll point out now that one of your workshops is on sliding filament theory, I believe, and that'll be a really important one to go to for that workshop. When the nervous system stimulates the muscle fiber, the myosin heads are allowed to bind to actin, forming that cross bridge, which will pull the actin by. So this is the cross bridge that forms, and what happens to each band and disc is described here. In general, many of them will shorten, some will disappear, but the point of the matter is the Z discs will slide past each other, not slide past each other, they'll become closer together as the sarcomere shortens. So this is a fully relaxed sarcomere of a muscle fiber, showing what you would see under a microscope. You can see that the darker bands will be where the myosin are because they're thicker, and the lighter bands will just be where actin are. So you can see this sarcomere again is from one Z disc to the next, and this is in a fully relaxed sarcomere, so a relaxed muscle fiber. There is some overlap between actin and myosin, but in a fully contracted sarcomere, you can see how there's much more overlap. The myosin globular heads are pulling the actin proteins past them, so we get a much shorter sarcomere. You can see here the Z disc to the Z disc has been shortened in width. So that is the sliding filament model because these filaments are sliding past each other. That's why it's called the sliding filament model of muscle contraction. Any questions so far? I know this is kind of heavy, but most physiology concepts are, unfortunately.
Professor, I have a question.
Yeah.
Is this the chapter 9, part A?
This is chapter 9, part A, and I'm sorry the PowerPoint isn't up yet because I ran out of space on Canvas.
Okay.
I will
Thank you.
probably by tonight.
Okay.
There's like a couple people still taking the exam who couldn't make it. So yes, I will upload this PowerPoint as soon as I can. I'm really sorry about that. I ran out of Canvas space. Okay, more on muscle fiber contraction. Decision to move muscles is always activated by the brain. It's transmitted down your spinal cord to motor neurons, which will then activate muscle fibers. Neurons and muscle cells are able to be excitable by action potentials. So excitable cells are capable of changing their resting membrane potential voltages. And that just means that an electrical charge can pass through them. An action potential is a word for that electrical charge that's being passed through these cells. And the action potential will cross from a neuron to the muscle cell that it's innervating via a neurotransmitter. So if you think about your nervous system is made up of nerves, your muscle system is made up of muscles. What's the connection between the two? Acetylcholine is the connection. This neurotransmitter, acetylcholine, often abbreviated ACH, will pass over a space from a nerve to the muscle fiber to cause that electrical stimulus to continue on to the skeletal muscle. Ion channels play an important role in changing the membrane potential or that electrical charge. There's two classes of ion channels. They can be chemically gated, which means they're only opened by a chemical messenger like acetylcholine. And there are specific acetylcholine receptors on muscle cells that will bind to that neurotransmitter. Or there are voltage gated ion channels, which will open or close in response to any sort of voltage or electrical change of that cell membrane potential. So this is an example of a chemically gated ion channel. So you would find one of these on skeletal muscle. It would have specific receptors that would bind to acetylcholine to help pass along that electrical signal. A voltage gated ion channel will allow ions to pass through. So the anatomy of motor neurons and the neuromuscular junction. So this will describe what happens at the junction between a nerve, a motor neuron, and the muscle. And we call that the neuromuscular junction. Skeletal muscles are always stimulated by what we call somatic motor neurons. Axons travel from your central nervous system to the skeletal muscle. Each axon divides into many branches as it enters the muscle. And these axon branches will end on the muscle fiber, forming the neuromuscular junction or the motor end plate. Each muscle fiber has one neuromuscular junction with one motor neuron. So this is just a look at how your brain sends out a signal through a motor neuron. And these axon terminals will end. The axon motor neuron will diverge into different parts of it so that each axon terminal will end at each muscle fiber. So each muscle fiber has its own kind of divergence of that axon of the motor neuron. And we call that junction where the motor neuron attaches to the muscle fiber, the neuromuscular junction. This talks about the axon terminal, the end of the axon and muscle fiber are separated by a gel-filled space called the synaptic cleft. Stored within the axon terminals will be synaptic vesicles that will contain acetylcholine, that muscular neurotransmitter. There will be infoldings of the sarcolemma called junctional folds that have acetylcholine receptors. So the neuromuscular junction will consist of the end of the axon, the synaptic cleft, the space between the two, and the junctional folds. And this is a look at the neuromuscular junction. So here's the axon of the motor neuron. The synaptic vesicles containing ACH, acetylcholine, are ready to be released into that synaptic cleft, the space between the two. And then you'll see these folds that have many of those acetylcholine receptors that will bind to acetylcholine. Once they bind to acetylcholine, then that electrical impulse can be transmitted through the skeletal muscle fiber itself. I like this slide because it just tells you the big picture of what happens. These are 4 steps that must occur for a skeletal muscle to contract. Everything that I just talked about, acetylcholine must be released at the neuromuscular junction. The muscle fiber itself must be excited. So acetylcholine will bind to receptors and cause that depolarization or excitation of the muscle fiber. Then the excitation contraction coupling. That means that the electrical signal will turn into a contraction. And then the cross-bridge cycling will occur. That means myosin heads will bind to actin, slide the actin past, and then release. So that's the big picture of skeletal muscles to contract. Here's another great picture of what I just talked about, but it takes you through in detail the steps of each. We'll start right here at step number 2. When acetylcholine is bound, it causes a depolarization of the skeletal muscle fiber cell, and depolarization just means that the membrane potential changes, and that means an electrical charge is going through it. The action potential travels down the T tubules, and that causes your sarcoplasmic reticulum to release calcium. Calcium will bind to troponin. Troponin was one of the regulatory proteins on actin. And when it does that, it'll shift tropomyosin, those spaghetti-like structures, off of the myosin binding sites. So myosin heads can then bind to actin, and contraction will occur via cross-cycling. So these are the events at the neuromuscular junction. And we talked about these, but this is a great slide for you to review the events at the neuromuscular junction. One thing to note, if there's excess acetylcholine that's been released, there's a specific enzyme called acetylcholine esterase that degrades excess acetylcholine. And this is important because if you have too much acetylcholine being released at this junction, you don't want to stimulate your muscle too much, or else it'll continue contracting. So this is a way for the body to control its contraction of skeletal muscle. Acetylcholine esterase will degrade any excess or get rid of excess acetylcholine. This takes you through the steps in detail. So again, use this PowerPoint to study it. I think these steps are great, and it shows you pictures of what happens for each step. This takes you through an animation of what happens at the neuromuscular junction. Some clinical kind of references to this: many toxins, drugs, and diseases interfere with events at the neuromuscular junction. So myasthenia gravis, which is a disease characterized drooping upper eyelids, difficulty swallowing and talking, and generalized muscle weakness. So kind of a grave muscle condition. All your muscles are much weaker, and it involves the shortage of acetylcholine receptors because a person's acetylcholine receptors are being attacked by its own antibodies. So this is an autoimmune disease. So that is an example of a disease that affects things going on at the neuromuscular junction. Generation of action potential. At resting, the sarcolemma, which again is the border of the muscle fiber, is polarized, meaning there's a voltage that exists across the membrane. We'll talk about this a lot when we get to the nervous system, but the inside of the cell will always be more negative to the outside, and the action potential is caused by changing that electrical membrane voltage. It'll occur in these 3 general steps. The end plate potential is when the acetylcholine released from the motor neuron binds to those receptors. It causes chemically gated ion channels in the sarcolemma to open. Sodium ions will diffuse into the muscle fiber, and this will result in local depolarization called end plate potential. Basically, simply put, sodium ions rush in, and this causes what we say a wave of depolarization. The wave of depolarization causes an action potential, or that electrical signal, to travel down the muscle. If you go to the workshop this week, or I think there's one tomorrow I'm teaching too, but if you go to the workshop, you'll learn about action potentials in the nervous system, and it'll help. We'll study that more when we get to the nervous system, but it will explain a little bit more what an action potential is and what depolarization and repolarization is. Repolarization just restores the resting condition. So basically, repolarization, after the electrical signal and action potential has passed by, repolarization takes that muscle cell back to normal. And these steps explain repolarization. This is a typical graph showing the differences in the membrane potential. So that, again, there's always an electrical charge inside the resting cell. Inside, it's about -90. And depolarization occurs when sodium enters it. So we get a more positive membrane potential. Repolarization occurs when potassium exits the cell, and that brings us back to a normal kind of resting membrane potential in a muscle fiber. It's a little bit different in nerves, but the same kind of idea exists. So go to the workshop on action potentials. Then excitation, contraction, coupling are the events that transmit the action potential, the electrical signal, basically to excite and tell your myofilaments to contract and slide past each other. And simply put, how that happens, the action potential travels down your T tubules, and your sarcoplasmic reticulum is in close contact with those T tubules, and calcium will be released when an action potential passes by. The action potential is brief and ends before the contraction is even seen. So this is a look at excitation, contraction, coupling, showing how the action potential travels down the sarcolemma and then into the T tubule, causing the sarcoplasmic reticulum, which is the lighter blue color, to release calcium. So here's the different steps. When the calcium ions will be released, this is kind of where the magic happens of the contraction. Calcium will bind to troponin. And again, troponin is this little protein in yellow. So calcium is required to bind to troponin. And when calcium binds to troponin, it removes the blocking action of the tropomyosin, those spaghetti-like structures on the myosin binding sites. So when we get tropomyosin out of the way, the myosin heads can then bind to the actin. It'll slide the actin past it and contraction will begin. This is another great animation of excitation, contraction, coupling. Again, when I put this PowerPoint on Canvas, you'll just have to download the PowerPoint in order to watch those animations. All right, questions so far? I know I'm not the best explainer, but this is one area of physiology I know well. So if it helps to listen to my lecture again, I would hate to listen to myself again, but if that helps, I will. These lectures are always posted on my YouTube channel as well. At low intracellular calcium concentration, so when there is not enough calcium in the skeletal muscle fibers, so it hasn't been released from the sarcoplasmic reticulum, that tropomyosin will continue blocking the active site on the actin, so the myosin heads cannot attach to the actin. The muscle fiber will remain relaxed until the voltage sensitive proteins in your T tubules change shape due to an action potential traveling down the T tubules, and that's what causes your sarcoplasmic reticulum to then release the calcium. At higher intracellular calcium concentrations, so when there is calcium present, that will bind to troponin. Troponin changes shape and moves the tropomyosin, the spaghetti, off of those myosin binding sites. And that will be when the myosin heads can then be allowed to bind to the actin, forming the connection, the cross bridge, and cycling will be initiated. And that will cause the sarcomere to shorten and the muscle to contract. When the nervous stimulation stops, so when there's no more nervous stimulation coming from your central nervous system, calcium will be pumped back into the sarcoplasmic reticulum to be used again, and contraction will end. These are the 4 steps to form the cross bridge, that connection piece between actin and myosin. In order to form the cross bridge, a high energy myosin head will be attached to the actin, thin filament site. The working or power stroke describes what the myosin head does. It will pivot to pull the thin filament towards the M line, which is the center of the thick filament. And in order for the cross bridge to detach, so in order for the myosin and the actin to detach from each other, ATP is required, and ATP will cause that cross bridge to detach. The cocking of the myosin head just means it's ready to get kind of back in action, to be ready to attach again. And this energy from the hydrolysis or the breaking apart of ATP will cock the myosin head back into the high energy state. And this energy will be used for power stroke in the next cross bridge cycle. The one important piece to remember here is that ATP is required to detach the myosin and actin from each other. So these are the steps of the cross bridge cycle. The power working stroke refers to when myosin heads pull the actin past it. The cross bridge detachment occurs when ATP binds and is hydrolyzed or broken apart to cock the myosin head back into its high energy state to get ready to attach to the actin again when calcium is present. Animation on cross bridge cycle. Rigor mortis. So I talked about this. If I had you for anatomy, I talked about this in anatomy, but maybe it will make a little more sense now. Rigor mortis occurs 3 to 4 hours after death, where the muscles begin to stiffen. Peak rigidity of your muscles occurs about 12 hours post-mortem. And this occurs because ATP is no longer being made by your cells. So calcium cannot be pumped back into the sarcoplasmic reticulum, and ATP is not allowing for cross-bridge detachment. So basically what's going on here, there's not ATP present and there's not calcium present. So your myosin and your actin are constantly bound together. So that causes this muscle stiffening of constant muscle contraction. Muscles will stay contracted until the actual actin and myosin filaments, those protein filaments, break down, causing the myosin to release, and then the muscles will then relax. So that's rigor mortis. So I'm going to stop the recording here.